Unlocking the potential of cell reprogramming lies in understanding the cells of the future and their role in reshaping cell identities. Greetings and salutations to everyone. In today's video, we will be discussing the fascinating discovery made by researchers from Helmholtz, Munich. They have uncovered a new connection between DNA replication timing, referred to as RT in the article, and the folding of genes into three-dimensional structures within the cell nucleus. Through the utilization of the Repli-seq technique, the team was able to create a comprehensive map of RT, spanning from the early stages of embryo development to the blastocyst stage. Surprisingly, the research revealed that RT in the single-celled embryo lacks a significant level of order, suggesting that genome duplications are highly flexible during this early developmental phase. These findings have the potential to revolutionize cell reprogramming by altering their RT program to a more adaptable one. Without any further delay, let us dive into our discussion. At the core of the transfer of life between cells and organisms lies the intricate process of DNA replication, which involves the duplication of genetic information. However, this is not a simple copying process. It requires a precisely timed sequence of molecular events. Recently, Professor Maria-Elena Torres-Padilla and her team of scientists from Helmholtz, Munich made a captivating discovery about this process, specifically regarding replication timing, RT, and its significance during the onset of life. The intriguing findings have been published in the prestigious journal Nature. The duplication of various sections of our genetic material, also known as DNA replication timing, RT, occurs at specific intervals. Scientists at the Institute for Epigenetics and Stem Cells in Helmholtz, Munich have utilized a method called Repli-seq to investigate the close connection between RT and cellular plasticity, which refers to the adaptability of cells. Interestingly, their research has also revealed a previously unknown correlation between RT and the three-dimensional folding of genes within the nucleus of a cell. Researchers have successfully mapped the process of replication timing, RT, from the earliest stage of an embryo, known as the zygote, to the blastocyst stage when the embryo implants in the mother's womb. Surprisingly, the RT in the single-celled embryo appears to lack order, suggesting that genome duplications are highly flexible during this early developmental phase. However, as the embryo progresses to the four-cell stage, the RT becomes more structured. This gradual shift in RT reflects the gradual accumulation of modifications to the DNA and associated proteins, known as chromatin marks, which indicate the activity and importance of genes in the cell's functions. Dr. Tsunetoshi Nakatani, the study's primary author, suggests that the recently uncovered information regarding DNA replication could potentially be utilized as a means of reprogramming cells. This notion is further supported by Maria-Elena Torres-Padilla, the corresponding author of the study, who explains that the early embryo cells possess an incredibly adaptable genome duplication program. These cells, known as totipotent cells, have the extraordinary capacity to generate every single cell within the human body. The researchers believe that their findings shed light on one of the factors contributing to the remarkable capabilities of these early cells. With this knowledge, it becomes conceivable to alter the identity of a cell by modifying its RT program to make it more flexible. The findings additionally indicate that RNA polymerase, which is widely recognized as the enzyme accountable for interpreting the genetic code and converting it into RNA, plays a role in shaping the specific RT program. This offers potential insights into how we might manipulate this program in the future. The team of researchers has uncovered that the formation of the genome's three-dimensional structure precedes the establishment of the RT program. This discovery is particularly thrilling, as it suggests that the manner in which our genome fits within the cellular nucleus's three-dimensional space impacts the adaptability of the RT program. Conclusively, the timing of DNA replication serves as a captivating element within the larger story of life. It reveals the interconnectedness between the accuracy of genetic replication and the ability of early embryo cells to give rise to various cell types in our bodies. Through ongoing exploration of these interrelations, researchers delve into a profound comprehension of the fundamental nature of transmitting life from one cell to another, from one organism to another, and the intrinsic qualities that enable a cell to generate a new organism. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.